So this session we will see about the molecular docking. Okay, so molecular docking, it's a process where you see whether the two molecules will interact or not. Okay, and how closely they bind each other. So that's the assessment that you are going to do by means of the molecular docking process. So there are certain steps that has to be carried out before performing the molecular docking. Okay, so the first step is, so you have already identified your target and what are the small molecules that has to bind to your target. So those selection of the protein and the ligand molecules is done. Okay, so next step is you have to prepare your ligand and protein. So what is done in the preparation step is you are adding hydrogens. Their valency is balanced and also the other minimization procedure is done. In that case, the higher energy of the molecule is reduced or it is minimized to an energy level where the protein is functional. Because in a normal in vivo system, what happens is the protein, they don't function in a higher energy level. For example, when you take a ball, okay, when it is in a movement, it has higher energy. When it is in rest, the energy is very less. So the proteins, when they are highly flexible, they cannot bind the other molecule and function. So it has to be minimized to the lower level before you do the docking process. So the protein and the ligand preparation is done by means of using the force field before you start the docking process. Okay, so after this preparation step, what you do is you do grid setting. Okay, so this grid setting is linked with the binding site residues. Okay, so you are defining the grid where your small molecule should come and bind. Okay, so this information only you have predicted from the CAST-P server. Or else, if the experimental structure, protein structure is already available, you can also get the information from the experimental structure. Because if they might have defined where the functional residues are there in the experimental structure that is solved by means of X-ray or NMR, from that also you can fetch the information. And in some cases, even the salt structure won't have the binding site information. So in that case also, you have to use the servers like CASP in order to find the binding site. So what are the binding site residues? In that site, the grid is set where your small molecule is going to bind. Okay, so with this PyRX software, which you are going to use it for molecular docking, the protein is not, that is, their flexibility is not induced. Okay, they are in a static conformation. Okay, only the ligand conformations. When you minimize the ligand, we get number of conformations for a single small molecule. Okay, so because in vivo, the proteins and the ligands are dynamic in nature, not more flexible, so that the energy is higher. But while binding, they will be having some movements. They will be dynamic in nature. So in this software, what we do is the protein is kept in a minimized conformation and the ligand is generated with different conformers with different energy level, so that the ligand will go and bind in a proper manner. Okay, so different conformations of ligand are generated in order to bind. Okay, so that's after grid setting, these all the ligands are made to dock. Okay, made to bind with different conformations to the binding site of the protein. Okay, after binding, you will check for the lower energy. So whichever is going to have the lower energy, that is going to bind stronger. So apart from it, you should the final step is you have to check for the interactions. Okay, because the molecules, they form certain interactions in order to have the binding with both them. That is, these are all not very strong forces. They are all like weak forces that forms the interaction between these two molecules. Okay, so what kind of interactions are made? Hydrogen interactions, hydrogen bond interactions, hydrophobic interactions, van der Waals interactions, okay, pi-pi stat interactions. So like this, interactions like weak forces, they form between the macromolecule and the small molecule. Okay, actually in the binding energy, when you compute the binding energy between the two molecules, these all the interactions are taken into account in order to compute the score, how these two molecules will come and bind. Okay, so these all the aspects only we are going to see in this particular session. Okay, so the first thing what you require, so already you have selected the protein and the ligand molecule, right? So that has to be loaded. So this is the PyRX tool that page. Okay, this is the environment. Okay, so here this page explains, that is this page, it is used for viewing the structure. Okay, and this is the what are all the files that are loaded within this software. So that information, it will be shown in this part of the window. And here the execution, the progress of the any the tools that you are running, so that will be shown here. Okay, so here you have tools like Vina Wizard, which you are going to do today, and this is the Autodoc, OpenBabel, Python. So these are some of the tools available. So their execution and the information will be displayed in this bottom part of the window. Okay, so now I'm going to load the protein structure into this particular window. Okay, so already we have downloaded the PDB structure from the protein data bank. So you are going to load that particular structure. So file, load molecule. So our target, what we are going to choose, is having a PDB ID 1E1B. So actually this is a cyclin dependent kinase protein, and this is also a cancer target in the humans. Okay, it is involved in the cell cycle. So we have loaded our protein molecule. So to use this to do some manipulations on the or to view the structure in different manner, you have to use left click in order to do rotation. Okay, right click in order to zoom in and zoom out, and you can use the scroll also to that is, if you are not rolling the scroll and if you are pressing, you can move the structure to any part of the window. Okay, and if you want to view only the structure without all these partitions, you can go in full view mode. So this is the option. Okay, and then you can operate on this structure. Okay, so escape will make you to regain the home page. So now we have loaded our protein molecule. So next we have to load our ligand molecules. Okay, so we have taken some ligands that is identified in turmeric, right? So that we can load it now. So you can use this OpenBabel tool within the PyRX in order to load your molecules. So there is a insert new item option which you can use to load your molecules. So it supports actually the SDF format files. The PDB is loaded in the that is PDB file format. Okay, the protein is loaded in the PDB format. So you can see that all the ligands that you have taken from the turmeric is loaded within this particular environment. Okay, so that is viewed here, and this protein molecule you can view it using 3D scene option, and here in OpenBabel window you can see the different ligands. So when you click on different ligands, you can view its structure. Okay, so this cyan color, it represents the hydrogen atom, and this gray bonds, it represents the carbon atom. The red one is the oxygen, and if some molecule, if it is going to have the nitrogen, it will be shown in blue. It is dark blue or color, it will be shown here. Actually in this ligands there is no nitrogen, Okay, so this is the color representations. Even in protein also the same thing. So this dark blue, it represents the nitrogen. Okay, red one oxygen, the gray one carbon atoms. Okay, is it okay? So our target and ligands are we have to prepare it before we go for the docking process. So how we are going to prepare? So there is an option within this tool. Okay, you have to select the structure. Okay, this is 1E, 1B. So you have to right click it. Using the autodock module, you have to make macro molecule. So it will prepare your protein. Okay, so it's done now. So this file, output file of the prepared protein, it will be saved in this autodock macromolecule. It is saved in PDB Qt file. Okay, so this is the prepared file. This is the loaded file 1E, 1V, and we have prepared and it is saved in this particular location. Okay, so next what we have to do is we have to prepare our ligands. So how we are going to do is we can select all the ligands. Right click. So you have the option minimize all. Okay, so minimize all. You have to first minimize all the structures because this ligand molecules, it has to be minimized in this particular protocol in order to do the docking. So minimization is done. So here you can see after minimization, you have the energy value. You have used the UFF force field actually for minimization. That's why you have got this, the name change. Okay, the ID of your ligand, PubChem ID of your ligand, the force field that you have used for minimization UFF, and their energy value. What is its energy value? So these are all the lowest conformation energy value of all the ligands. Okay, so different ligands will have different energy minimized value. Okay, and after that you have to prepare your ligand. Okay, so that is done by converting all to autodock ligand PDB QT. So this option has to be used for ligand preparation. Okay, so now all your ligands have been prepared. So you will have all your ligands in this particular folder. So before it was not there. After you minimize it, it will be saved under this particular window. That is a folder. Okay, all the minimized ligands, it will be saved in deep. So here it will be having the energy, minimized energy, prepare the ligand is available here. So we have prepared both the protein and the ligand. Okay, so now we can go for the docking. You can start the process. Okay, for that you have to choose Vena, Auto Vena Wizard. Okay, in this you have to give the protein, prepare the protein as input and prepare the ligands as input. So for that, so first you have to start, and the option here it should be local because you are going to use the execution file Vena from the local folder only. So here you have to select the molecules. So in the select molecules, add macromolecule option, you have to check it, or you can we have in our window itself the prepared protein. So you can choose it and give add macromolecule. Okay, we should select, it is getting added. So you can see here, Nyan ligand selected, and your protein file is also, the selected ones will be automatically taken for the docking process here. Okay, so the next one, forward, the next step. So this is the grid setting step. So here you can see the dimensions. So this is the 3D space where you are viewing your structure. Okay. So each atom has 3 coordinates, that is x, y, z coordinates. So the dimensions of this particular grid. So you can see when I change the grid size, here the x, y, z coordinates are also changing. And the center, it is in here. Okay, where you want to center in this particular protein structure 3D space, their coordinates are given here. Okay, and you can either maximize, minimize, or you can alter it. Okay, so in the 3D space, so we have the binding site information, right? So now you have to use that information in order to set the grid in that particular binding site. So we have already the residues 194, 198, 200 to 204. Okay, so these residues I'll take it as the information in order to select the residues within this particular 3D window. So you can see here. So what are all the residue numbers? So 194. So this I will make, because you can see here how all the residues are distributed. So better to label those residues where those residues are present, and then that you can use as a clue in order to set your grid. Okay, so where is your binding site now? I'll maximize it to full screen mode. I'll move my grid towards to this region. Okay, so now I'm using left click. Okay, so here I want to rotate and see, because in 3D space, the objects that you see is not exactly in the same position. Okay, so you have to set it in a correct position so that your ligand, whatever you want to dock, it binds to that particular position. Okay, now the grid is set. So for the publication, you might be that is, you are required to submit these informations where you have set your x, y, z coordinates and what is the dimension of your grid. Okay, so next we can go for forward, which will, so here the exactness is 8. Okay, it's an algorithm where it will use this value for docking your molecules in the binding site. So forward. So now this is the docking process. You can see the status, which molecule is being docked. So number of confirmations. Okay, now we have given only one confirmation for docking. It will search for the different confirmations where the binding energy with interaction is happening. Okay, that is the docking, that is auto-vina it will do. Okay, just it will search in which confirmation the small molecule can go and bind in a that is in a proper minimized conformation so that their interaction energy is very less. Okay, so all those are fetched in this particular process. Sometimes after getting the result, you may get for a particular ligand, you get different conformations with different binding energy value. Okay, and one conformation it may have the lowest energy, that is -11 or -12 like that. But it may be anything, depends upon the ligand that you use. Just for example, I am telling, if the lowest conformation, sometimes it will not have interactions, hydrogen bond interaction at all. Okay, so while checking for the interactions, you have to balance both the energy and also the interaction when you find out the best conformation of the ligand that binds with the protein. Okay, I'll tell you, I'll explain you in the next process. So still some 3 more ligands are to get finished. Then the next step will be, you will be using Discovery Studio Visualizer in order to find the interactions between your protein and the ligand molecule. Okay. So you can see the tabs here. So 3D scene, actually it is used to see the 3D structure of your protein, macromolecule whichever is available, and the process which it runs, it will come in this particular tab. Okay. So here you have the documents option. Even you can open the document and see the minimized energy, that information is also, it is available as a tables. This particular target docking results, this also can be viewed using this particular option. And you can also save this information in a CSV format. If you want to save this minimized energy, if you want to have this, already it will be named all the ligands and the protein files, whatever you are using in this particular window, it will be saved in a particular folder. Okay, the folder is, it will be automatically creating a folder and save all these things. So in a PC, in the C drive, users. So whatever the user you are in your system, whatever name your user is, .mgl tools. So I have not created any folder before starting this session, right? But it will be automatically saved in this particular folder only. All the information, the prepared protein information, prepared ligand information, so everything will be saved in this. .mgl tools, PyRX. So in this you have the information of ligands in this ligand folder. All the minimized energy files will be available in this particular folder. All the macromolecule protein information, it will be saved in this particular folder. So here you have the ligand files also, right? So these are the conformations of the docked ligands. So this is the folder, ligand folder. It will give you the information of the prepared ligands, not the docked ligands. Okay, whereas this particular folder with the protein, your ligand molecules, it will give you the information of the docked ligands, docked small molecules. Okay. So now the docking is finished and the energy values are reported. So you'll get the results in this way only. Okay. So the protein name, the ligand name, the whole, all the 1, 4, 3, 6, 7, 5, 5. So all these are ligand name. Okay. As far as how we are giving the ligand name, the same thing will be taken in the output also. Okay. Next thing is the binding affinity value. Next is the mode, RMSD lower end and RMSD upper end. So what do you mean by this? So this is the binding affinity, which is based on how far the small molecule that we have, ligand molecule we have given, how far it is binding with the protein molecule. What is their energy value? Binding, that is how far it is the strength of the protein with the ligand. So that is defined in terms of the binding affinity score, and the unit for this has to be mentioned as kilocal per mole. Okay, and what is this RMSD values are? It means that, so when you are giving the ligand in one particular conformation, so when it is trying to bind in different conformations while you docking, okay, there will be change in the RMSD, root mean square deviation from the conformation of the ligand you are giving and in which conformation it is going and binding. When it explores for the different binding region, there is different conformation of binding, the RMSD change will be observed. So that RMSD value is given here. Mostly the ligand will have the lowest binding affinity value only with RMSD, that is the minimized conformation, it will have the lowest binding affinity value. Okay, so you can see here for each ligand you get many values. The first one is always having the lowest binding affinity. Okay, here the -6 is a binding affinity, lowest binding affinity for 1, 4, 3, 6, 7, 5, 5 ligand. Okay, and this ligand 18818, it is having -4.9 energy value, and you can see here for this also the energy value is, that is the RMSD value of upper bound lower bound is 0.0. Mostly the lowest one will be 0 value of RMSD, and the binding energy for each of the ligands you can see here. Okay, it is distributed in this way, and there are options that like Excel options, sorting is possible with this particular tool. So before that I'll show you this particular, all the results, it can be exported in a CSV comma separated file format. Okay, so that is can be done, save as comma separated files. So you can save all the results in a Excel file. Okay, and so when I click a particular ligand, you can see in the protein, okay, where the ligand has bind docked. Okay, you can see here. So the conformation change. Okay, third conformation, fifth, so even fourth is binding in some other. So mostly the first, that is binding energy less, will be mostly binding in the same region. Actually, you can see we can see two ligands here, right? Actually, this is a ligand which is found in an experimental structure. That is, experimentally they have identified where the ligand will bind. So that one, you can see the first conformation predicted is in the same position it has predicted the binding. So that's the power of the docking tool. So you can find the conformation, proper conformation in which the ligand molecule. So when you are doing some experiment and you are obtaining many biomolecules, so if you want to screen or if you want to choose only a small set of molecules that you can take for the experimental testing, that's why this docking will play a more important role in finding those molecules, filtering only those molecules which are having higher affinity. So you can see here among the ligands what we have given, You can see this is the ligand which is having the lowest, that is highest scope, binding strength. Okay, lowest value, that means is binding well. Okay, so this is the ligand when compared to this particular ligand 18818 or 4431. So all the values are less when compared to. So when you want to choose only one ligand among the 7 ligands or 9 ligands what you have chosen, what you will choose? You will choose the this particular ID 1436755 with this confirmation. Okay, now from this exercise, for me, the best ligand with the lowest binding score or the high strength is this particular ligand. So now I want to save this particular ligand in this confirmation. So what I can do is I can save this in a SDF format. SDF is a, you can save the three-dimensional coordinates of this particular ligand in this particular confirmation. Okay, save. So in the desktop, I will save it as doc best confirmation molecule. Okay, I will save in the desktop this molecule. Okay, so now what is getting saved is only your confirmation of the ligand. Okay, you are not saving the entire protein with the ligand complex. Why? Because the protein we have not changed the confirmation. The same confirmation what we are giving is used for the docking process. Okay, so the input file whatever you are giving, it can be used in order to analyze the interactions also. So now I have checked the protein and also the ligand in which the best confirmation or the possible confirmation with the lowest energy I have found. Okay. Now I want to analyze the interactions. Interactions, it is best to view with some of the softwares like BioV, Discovery Studio Visualizer or PyMal, Chimera. So those softwares can be used in order to understand the interactions between the protein and the ligand. Okay. So now we have the best confirmation ligand molecule. Okay, I have saved it and I have also saved the that is binding affinity values of all the ligands in the Excel. Okay, so these are all our output files. Okay, so now I'll minimize this window. I'll check the interaction. Okay, so now what I have to load, I have to find the interaction between the protein and the ligand. Okay, so now I'll load my protein molecule. So this is my protein molecule and I have to load my ligand now. So which I have saved in the desktop as doc best confirmation. So it will open in another window. Okay, so this is the structure of the doc confirmation. So what I can do is I can just copy this coordinate. Control C. I'll select this. I'm just double clicking the molecule. I'll keep the cursor on the ligand. I'll double click the molecule. Control C or right click, copy. And I'll paste it here. Okay, so what I have to do now is I have to select the ligand and I have to find the interactions between the protein and the ligand. So already there is one ligand which is present here. It's an experimental solved one. Okay, and this is the ligand which we have found. Our ligand in best confirmation where it will bite in the same binding site. Okay, so for clarity, what I will do, I will delete this. So you have the protein and the ligand molecule now. So you can see the helices, sheets, loops, everything here. So now what we can do is go for the tab receptor ligand interactions. Okay. Now the receptor or your template, target protein, it is name is 1E, 1V. Now you have to define the ligand. You have to select it and choose here. So it will get selected. So your protein is selected, ligand is selected. Now you have to go for the ligand interaction option. So where you can find the interactions of ligand with protein. Okay, actually what happened is only the interacting residues are visible here. All the other residues are removed from this window when you use this option. Suppose when you want to view this, so you can first, the thing is you want to know what are the residues that are interacting with your ligand, right? So you can add the label. Select, add residue name. Okay, which color you want to choose, what should be the size of the font which has to be displayed. So everything will come to your, it's depend upon your choice. Okay, apply. You can see here. So the molecules or the residues that are interacting with your particular ligand. So this is your ligand molecule, and if you want to change the color of this particular ligand, display state, you can choose ball and stick. Like, okay, you can differentiate the residues from that of the ligand. Okay, and I want to have my protein structure also here. Okay, so what I can do is what I have done is I have selected this particular option. The protein, this A chain, actually it's only one chain is present, A chain. I have selected it in order to view that. So you can view the protein ligand interaction with your protein. Okay. So there are different colors of interactions, right? Pink, green. So this also has its own meaning. So you can go for the option show 2D diagram. Okay. So there you can get the meaning of each of the interactions. So green color, it is a hydrogen bond interaction. So this with the pink color, it represents the alkyl interaction. The blue ones, that is the ligands. So in vivo, the protein and the ligand only doesn't exist. They also have water molecules, right? So in the structure we have loaded, there is water molecules also. So our ligand, it interacts with the water molecule also. This also play a more important role in stabilizing the interaction between the protein and the ligand. So we can also consider the water molecules that is interacting. Okay, so these red colors are actually unfavorable interactions. Okay, so this blue, it represents, sorry, the green, this light green, it represents the van der Waal interactions. Okay. Actually here, You can also remove, suppose these are all actually the unfavorable interactions, the water molecule that is present, right? So you can also remove it by means of changing the options. So here, actually it is default what are all the interactions you have to view. So those options are given here. Suppose if you don't want to have the unfavorable interactions on your window, okay, you can choose this option. That is, you can untick this option, apply, okay. So unfavorable interactions are removed. Okay, this way you can do the docking and you can analyze the interactions. So when you discuss in your article or publication, you have to tell what are all the residues that are in the binding site are interacting with your ligand, with which atom in your ligand. That is also important. So you can see here the oxygen here, it is forming a hydrogen bond with the glutamine, that is 131th residue. Okay, so this has to be mentioned in your publication. And what are all the other residues that are interacting and which group in from the ligand or atom from the ligand. Here it is aromatic group that is forming the pi shaped interaction. So like this, what atoms within the ligand are also interacting. That also is very important in a publication point of view. So you can have this picture in your publication, just you can copy and paste it in your PPT or in a Word file, and you can have this, or else you can also have the save as option. Different image files you can save, which you can use it for saving your interactions.